Okay. Dagen, thank you so much for this careful analysis. I think, as I said in the email, this is all the things that you found, and all these observations are are a new project. Yeah, there are so many questions. So I started writing an email, but it was getting too long. So I'm just going to tell you something that I think. So, for example, I think this is very interesting that you are saying that some of the tumors, these CTC now, let's call it like that, the CTC here and here, they don't come from only from tumors, but they come from our regions that we cannot see. So they appear in the image. So they can come from other legs or from other pieces of the tissue that we don't know. So yes, this is something that we can include in the paper. That actually the majority of the things that we see are not breaking from the tumors. They are coming from places that from other parts of the image that we don't know exactly where they come from. That is important. Um, These here, you are talking about the appearance and disappearance, that they go from 1 into 4 or 1 into 2. That is a great observation. And in particular, I don't know, in all the movies, like in maybe in the movie from Sonia, this doesn't happen very often. No, I think in all of the movies, maybe this happened that unfortunately we have a lot of pieces of all these CTCs that are in one slide, and in the next one they are lost, and very few are the ones that we actually can continue following. This could be for two things. One, remember that we are using a technique that is called confocal microscopy, and these take pictures by layers. Yeah. So one thing that it could be that one of these spots could be in the layer below when you went to the bottom. So if we take maybe the next semester the original data, we can see if when I fuse all these pictures, maybe they are in the bottom or they just disappear. Yeah. So if they can be in another region in the picture, or maybe the second option is not that they are on the top or on the or below in the bottom. The other option is that they are moving much faster. Yeah, and remember that I think each picture that we took is every 10 minutes, so we will need to use, instead of confocal microscopy, we will use another a different type of microscope. The what I use is a regular microscope, a regular confocal microscope. There are two different scopes that are much faster, so that instead of taking pictures every minute, you can take pictures uh, every 2 seconds, every 10 seconds. Yeah. And this I will send you the names. One is the an spinning disk microscope, that it allows to take pictures much faster, so we can see exactly where we're losing the spots. Or there is another one that is called light sheet microscope. I will look into that, and I will send you the names. So the idea is that maybe we can write in the paper that one disadvantage of this technique is that the tumors can move very fast, so we don't know what happened with them, and we cannot follow them. So we should maybe explore with other type of microscopes, like spinning microscope or the other one that are new microscope that they take pictures much faster, so we can see what is happening. Maybe we're missing the attachment and or the detachment and reattachment of our cells. But and if we use those other microscopes, maybe we can see those. So next semester, we can see these things, and in the paper you can write about that. Mm, Again, this is the same idea. So that just write in the paper using another type of microscope and just describe that not only we are seeing tumor spots coming from our regions, but we see some of them appear and disappear, and it could be because of those two reasons. They could be up or below of our stack of the picture that we took, or they could just move much faster than we anticipate. And this is example one of the example of the trajectory that we can follow and that we can predict. And this is one of the main results of the paper, that although they are in different groups and they do different things, they move in same always from proximal to distal. And if I understood well this analysis that you're doing here, uh, this is another thing that is very interesting. This, when you say that is cell division and reduction of size, here this image, Sonia also have that thing, and I don't know exactly what it is. Maybe if you read the papers, one tumor that she found that it detached, that it broke from another piece of tumor, had that shape. So Sonia also have it, and I don't know exactly if what it is. We don't have. We will need to have a special marker to see for cell division to test it, and um, I don't know exactly what it means. So, but it's something atypical, and it was found before, and I think it's important to say that it was saw it in the in the tissue that Sonia also of that broke. So generally they have these regular circles, but this one have like a it's not as regular, so that is interesting. Red tumor, so it reduced in size, so it could be, it's interesting, it could be dying, or we just it's going to the bottom of the slide and we only see just a tip of the cluster. So we just have to next semester go and try to see in the original data and see what happened. If before we were imaging in the center, so it was the of this CTC of this cluster, we are just uh, we of this circulating tumor, we were just imaging the center in the beginning, and later in the next slide we were just getting the bottom or the top, and this is where we are missing, is reducing in size, or there is actually a cell, a behavior that the cell is getting smaller for some reason. So it's something that maybe you can discuss that in one or two sentences. You can say that we use different. It's necessary to use different microscopes to see, for example, to have a better understanding of these behaviors that we don't know exactly what is happening. Yeah, like the movement, that they move so fast, that they move from other regions, and in this case that they seems to get smaller. So in one or two sentences, or if you find something in our papers that they have described this, we can discuss a little bit. Mhm. Uh, Disintegration, tumor spot pulse. Okay, that is, yeah, definitely talk about this. Then we talk about this in the uh, in like a, try to see if you find it with a different name. We call it like a the attachment, I think, or and reattachment when they join another one. And Sony also found one like that, that the tumor break. Yeah, so talk about that and said that we need, if you remember, that we need to analyze that in more detail, take the raw data, and maybe have two different fluorescent markers. One red and um, one green, one red for the leg, and one green for the tumor. So we clearly see that it's breaking from there, and we can see it if it's able to actually attach to another place. So yes, that should be in the paper. And Randers said that definitely we found this. I think what we should say is that this is an interesting behavior that it seems to be very important, and it should be analyzed in more details, and try to propose techniques that how to have a better resolution of this technique, or of this behavior that is very important. Okay, this is the same thing that, yeah, it's just very interesting. I think maybe if we check in a lot of detail, I think Levi also found something like that. That we have two pieces of tumor that seems to be detaching. Yes, so we said that they seem to be detaching, but and as I said before, that we should check in detail what is happening. Brian, that completely say this is true, and yeah, you find, so it didn't happen only once. It could happen multiple times. Here you show that. Here you find another one, and again we have to go on the next semester. So in the paper, just say that they seem to be doing this and suggesting how to have a better resolution to see these things, having a different microscope, having suggesting using two fluorescent markers, or more things that you imagine. And later next semester we can go and see the raw data to see if actually uh, they are in the bottom or in the top, or they were never there. So we find that we can have definite data, a conclusive data of this is happening, if this is happening or not. So just keep that for the next semester, and we can analyze it. And I think that Levi also have one close to the wing that it also seems to be doing something like that. Yeah, we have to, again, same thing. Yes, to go next semester and see that and only suggest this. And this is very interesting. Same thing. We just have to go to the raw data and see, and see if they actually were able to leave the epithelia or not. And maybe even with that we are not going to have enough information, but this is something that we should document, that all these behaviors, they might be happening, and we should find different ways to test if they are happening or not. Yeah, that is next semester we can check that. Yeah, and this is also very interesting. We In the beginning we talked of some cells that are coming into the image, into the region that we are imaging. So they're coming from here, but there are also some that are living, and they generally live in this direction. So this is something that we should also report, and that it should be studied in more detail. Okay, so those were amazing observations, and maybe later uh, we can talk more about it.